Thank you. Why don't we get everybody from the group to come sit up there and see if we have discussions for the group. About 30 minutes. Okay, we have an audience that's bravely sat here all day, but I'm sure they still have some inspiring questions. Maybe. Questions for our group?
I wanted to say one thing.
Okay, speeches.
I think, no, it was in response to the question about atopic dermatitis, and correct me if I'm wrong, but I think that one measures the permeability barrier of skin, it's done using a device that can monitor water vapor. The simpler way to do it. You see that even on unaffected skin of a child or an individual with atopic dermatitis is a problem. So there's a fundamental problem in the barrier function. It only is intensified by the co-infection, by the infection of Staph aureus. Is that fair? It's a fair statement. Okay.
Yeah, I mean, there are mutations in the skin barrier protein filaggrin that are the strongest genetic association with eczema, and that means that the child's not building, the normal skin barrier. It's very interesting that those mutations in filaggrin are strongly associated with eczema that progresses to asthma or allergic rhinitis, even though filaggrin is never found in the lung epithelium or in the nasal epithelium, suggesting really that it's sensitization through the skin and then re-stimulation so that there is some, there is some connection. People had always thought that that meant that there was an immune, that was driven by the immunity. But in fact, if it's related and it is very strongly associated with a skin barrier protein, that means that it is something about the initial antigen presentation through the skin that is remembered in terms of the allergic airway inflammation and the nasal epithelium.
Dr. Fletcher.
I'm on shaky ground here because I'm a plant person, but I wondered about other kinds of, it's sort of related to the last question, but it has to do with allergic reactions that produce a rash on the skin, and my understanding that these can be sort of a systemic thing, but I'm wondering if it's related in any way in terms of the skin reaction, and if the condition changes the microbiome on the skin.
So contact dermatitis, I mean, there are people, or plants, like the way people respond.
No, I'm talking about an allergic reaction to, like hives. Yeah, okay. Like hives.
I haven't seen studies with a microbial involvement of that. There certainly are a lot of these kinds of triggers that are unknown origin. And I just haven't seen any studies that are looking at, the microbial diversity of classic urticaria or, those kinds of studies, and whether people have thought about them as a microbial. We talk about that as host environment, but really when we talk about that, environment is often the microbes. And, where do we put them on the gene-environment interaction scale? Are they part of our genome, or are they part of our environment? And I think that's part of, even the group discussion is to claim this as perhaps more part of our genetic component, but much of the plasticity is like you see with the environmental triggers, that they can change more rapidly than our 23 chromosome genetic state. So I think that, because clearly that's something we could modify, and I think, again, even some of those will happen during a certain period, during, childhood, adolescence, and then kids will grow out of them. And that's also something that, as you say, like when you think about eczema, is it that someone's microbiome has finally set, and now they're not going to have these modulations, or the classic urticaria, the hives? I mean, is it that finally your microbiome set, and you don't have those fluctuations?
Yeah, I just wanted to comment on Michael's last story, and just to say that it's one of those stories that just addresses the idea that nature and evolution has done phenomenal experiments from which we can learn. And I think something that humans forget on a daily basis is that we are part of the environment, and we are very, highly linked, despite the fact that we separate ourselves from it so well. We're part of the environment, and more importantly, that we are products of our evolutionary history. In our evolutionary history, all that diversity that's out there, if we understood the very basis of what goes on, these comparative approaches ought to give us some really great ideas about the very basic rules by which animals and plants interact with microorganisms. And we know this has gone on since the beginning of the eukaryotic transition. And so, just a comment.
Can I since I'm sitting here now and haven't included this formally, this is part of my musing. The dolphin is phylogenetically related to an ancestor of the hippo. So an animal that, but an animal different than the hippo, dolphin. And if you go back to man, vertebrates, the healing of that animal is very reminiscent of the fetus. So one of the thoughts I've had, and it clearly had to evolve in a very interesting way, and one of the thoughts was that perhaps there's a program, that we have that sort of switches when a program with a big, involving a big jump of a lot of different genes that is activated when we go from fetal, marine, to terrestrial. And maybe the dolphin, which had to evolve in the waters of the shark, which preceded it, retained some of those fetal programs. And what we're looking at in the dolphin is a fetus.
It's a derived condition. It's a derived condition because they are a derived group, crown group. But it would be perhaps retention.
Retention,
with,
yeah.
The neoteny.
Neoteny,
yeah.
That's exactly what I was saying. Exactly.
Thank you. Caroline Smith, DeWall Center for Science in the Public Interest. We do a lot of work in translating science like this back to the public. And a lot of the public's very germ-phobic, and so some of the interesting information coming out in the last 2 days has been about this blanket of protection that the microbiota may be offering us. But my question is, I heard a couple of very interesting questions, Julie, one in a question you asked earlier, but also in the presentations about the role of hand washing. Hand washing is a key consumer message in dealing with food safety, in dealing with disease avoidance, norovirus, for example, or flu. So I guess I know this is developing, but I would be very interested in your thoughts on what our advice should be to the public in terms of hand washing. Thank you.
They're doing fecal transfer.
I think this is, so there is a great divide between Activa yogurt and Purell, and so in this country we want to colonize our guts and sterilize, our skin. So I am a big believer in hand washing. I was even struck. I think there is. There was a recent article in the New Yorker that was all about hand sanitizer. So and how hand sanitizers have changed. So it's definitely very much on Americans' minds. Is every European who comes over to visit me is to shocked that there are these Purells in every elevator well. So this is an American. We have produced this need, but I think there is a distinction between, yes, you need to wash your hands, especially, we are concerned about oral fecal transmission. Full stop. That is really what we need to be worried about in terms of the normal human healthy, and that's different than an ICU. But yeah, for an oral fecal transmission means that you are washing your hands after going to the bathroom before you prepare food or eat food. The thing is, if we could get people to do this, I think the message is clear. And now it's about making sure that people really do it. And, flu season, I don't know that I have anything new to say other than, yes, people should wash their hands.
Yes, I agree. Also from Europe, yes.
Yeah.
With Purell, too?
I used to be the crazy mother who was going into telling, the teacher that my kid couldn't do this and couldn't do that, and she couldn't, because the whole pump thing. And I think that the messaging is all wrong about, killing the bacteria and so on. But if that is now going to be that you're going to get better adherence, people are going to use those hand sanitizers. I would like this to be evidence-based. But in the meantime, if Americans cannot wash their hands but can use Purell, I'd rather have them using Purell than being unable to wash their hands. So I think it's a cultural messaging thing, but I'm not opposed to it if people then develop this and they can do it 99% of the time and they can only wash their hands 47% of the time, fine. I don't use it, but, I like to rub between, the fingers of, the digits of my finger.
Yeah, I think if there's anything coming out of the science, it's that the paranoia of, that all germs are bad and that somehow we can totally control an environment and that will be a healthy thing to do, needs a major reexamination. And, it's been very unnerving to see, for example, parents getting advice that children should, have no contact with anyone for months and things like that. And I think this kind of mentality could be harmful and is very different from basic hygiene that can save lives. But I think as the science emerges, it's going to be interesting to see that. Dr. Kester.
Well, going from hand washing to fecal transplantation, so it occurs to me that I think the fecal transplantation. It's a great observational type of approach, and yet I think there's so many unknowns. We've heard a number of talks where sort of the stool contents have been fairly reasonably well characterized in terms of the predominant microbial flora. Could we envision a time, instead of doing just sort of, kind of brute force fecal transplantation, that we have standardized culture inoculum of, like, the top 5, of organisms, and could that be just as good, better, or would that cause other problems?
No, I expect that fecal transplantation is a temporary solution, and that I think 5 bacteria that will probably not be enough, but somewhere between 10 and 30. If you make the right mixture, you will probably be as effective as transplanting the whole bunch of bacteria. But much work has to be done there.
Like maybe you could have like sort of standardized sort of bowel reconstitution package of.
Yes, after every antibiotic course, exactly. Especially before age of 3 years, maybe. Yes, right. Yeah. Probably that. I think the easiest way to develop such a mixture of bacteria for Clostridium difficile, because there you have a true clinical endpoint and you can cure the disease. So you can. That's the model to start with, and probiotics. That's just kind of well, it's kind of lottery. You don't know what you need, and you just give some of it. So that's my opinion about the current treatment with probiotics. But probably you will have in 10 years or something probiotics that may work for IBD or pouchitis or things like that. And yes, I think we can expect a lot of it, but we are not. It's just speculating.
I think we can also get there in other organ systems, so like skin. I mean, I think that there's a possibility there of delivering, it may be live bacteria, but in this country it may be, small molecules that promote the growth, or even, the creams that we use. I consider them probiotics, that they're fertilizing the microbial garden. So I think it's not just only about prebiotics. I don't think it's only about, fecal transplant. I think that this could become a more common theme for treating other body types and restoring the microbial diversity.
It's also interesting, though, that we're taking a really medical approach even to the microbiomes. I find that fascinating. So it may be that we learn more preventive approaches to not disturbing it to the degree we do, or to restoring it, rather than, you have a disease of your microbiome, therefore we're going to give you a treatment. Yeah.
And so lots of discussion over the last couple of days about antibiotics, with no discussions over the hundreds of thousands of tons of broad-spectrum antimicrobials that are in all of our personal care products. I don't think anybody can find a soap or a shampoo that doesn't have paraben in it as a antimicrobial preservative, and things that we take into our mouth, food additives. 700 items on the FDA generally recognized as safe list. I don't think the bacteria had much of a vote in that. How is our changing American, Western, increasingly global diet affecting our microflora in the gut with just no awareness that's out there? Maybe some of that variability that we're seeing is reflected in their diet.
Can I make a comment on, and I'm going to ask Julie this question. I did a clinical trial on, and this relates to your point, that infected diabetic foot ulcers without, and this was a 1,000-person trial, and part of the FDA's requirement was doing, the FDA doesn't really care about the microbiome, frankly, and it shouldn't in the treatment of an infection. They want to know if the infection is cleared. Then they worry about the things you measure. They just want to know, is the patient getting better or not? Is the wound closing or is it not? And one of the things that was really interesting was that I think that if you take that system you're describing, the body's topographical, or the distant sort of geographic, microgeographic clusters of colonies of whatever, of organisms, this map, and you take an antibiotic, let's say a topical antibiotic, and you clean it off, right? It's been my experience, at least with this population, that things just go back. It's as if you put your hand in the ocean, and then you separate it a little bit, and things just goes up again, and there's a sort of buffering. The organism spreads. The organisms sort of spread or disappear, and then they creep back. You follow what I'm saying? So let me ask you a question. Have you done a study, advice, in which you've taken an area, let's say the antecubital fossa, you put some antibiotic on, and then you let the system restore? What comes back?
So the bacteria are deep into the hair follicles and the glands. So cleaning off the skin superficially is not depleting the microbial communities. I think that Brendan and the ecologists will talk about the transition of biotomes between you have this microbial community and you have this one, and how do they at what point do you see, is it a sharp transition? So I think all of those questions could be addressed with, human clinical trials. I guess the question is, I don't know, if you can come up with, I need to know this for a medical reason, I will do this trial. But I don't really know, that's an ecological question, and it's interesting, but to me, is it about, if I put a cream here, I'm going to get a field effect, and I'm going to get, something else here. I think your question was about that now people want to leave a pot of, cosmetic cream in their bathroom and not have it turn pink, and they want to leave that pot there for a month, of hand cream or something, and the company, Procter & Gamble, does not want the phone call that says, my cream turned pink, and, I've been putting it around my eyes, and now I have an eye infection. So, I mean, the marketing of products that are used, food care, I mean, there's a lot of antimicrobials that are going into the whole system, and I think that is something that as we think about, it's not just antibiotics that humans are taking, it's all those antimicrobials that go into the fabrics that are used to construct couches, the antimicrobials that are going into foods, and and you can't talk about any of this without the amount of antibiotics going into livestock. But there are whole forums that address that. And it becomes sort of the system. We have, all these inputs and all these outputs, and each one of them is incredibly important. So I think that you've asked a question that's really, an entire forum discussion.
But it's a very, in a way, when I hear you speak, I sort of, fear, you get this frightened, this sort of fear that is emerging that, we're killing ourselves, we're destroying ourselves through the use of antibiotics, and I think that's sort of going a bit far. The paraben issue, I mean, there are issues that relate to that, and I don't really know the answer, to know the extent to which they actually are effective at the concentrations in which they're present in these various organic formulations is not clear to me. I don't know how effective they are in the skin. Do you? No.
And that's the issue.
Well, many of them are they're organic, they're not terribly water soluble, so it's not entirely clear what their use, what their efficacy is as a topical disinfectant. So to generalize and say, antibiotics use is dangerous. As you said, the use of preservatives really has to be questioned. Yeah, absolutely, but I think it should be done from a position of not creating fear. These are reasonable questions, but at this point in time, I don't think many people who worry about it are really worrying.
Marty's going to answer that.
Marty will say he's measured it and I'm completely wrong.
No, I think the problem is that and there are many antecedents to the problem, is that everybody is germophobic, and the thought is that all these manipulations don't have any biological cost. There's no biological cost, long-term cost to giving antibiotics. There's no long-term cost from paraben or triclosan. And we don't yet know, but I think if we just consider, if we develop the a priori hypothesis that these powerful antibacterial agents may have some role in perturbing our ancient microbiota, then a lot of things follow. Now, the experiments have to be done. The epidemiologic studies have to be done. The clinical studies have to be done. The animal models have to be done, but they're being done. And to say that there's no cost ignores the fact that we have all these diseases that are rising, all these epidemic diseases for which one can make a plausible argument that the microbiome is involved, that perturbation of the microbiome may be involved. These are testable issues. But I think we should go back to the meeting of this forum in the book from about 10 years ago, Ending the War Metaphor. The public is still back in the war against germs, and we have to educate the public better, that there are good germs as well as bad ones. Mark Allard, I'm with the FDA. I just wanted to put the other side of it, and that is that Food and Drug is responsible for the safety of cosmetics, and the public expects safe products. And so that if you put eye wash in your eye and you get a fungal infection, they want us to remove that. And the FDA is giving a lot of scrutiny to this whole industry, and that's the other sort of whole story that has to be balanced.
Thank you. I mean, yeah, we do benefit from a lot of infections we no longer get, and we do sometimes see with medical progress infections we do need to deal with, like amoeba in contact lens solutions. So, other question?
Well, I don't know if it's a question as much as a comment. The compounds you're talking about are actually preservatives that are used to extend shelf life. And so in many ways, these might be likened to low-dose antibiotic treatments. I mean, no one's going to put these things on to treat an active infection, but they are used for chronic preservation. But I too have been struck by the germophobic nature of the public. As a microbiologist, it's really quite striking to hear people talk about germs instead of bacteria. But the people that are marketing these hand sanitizers are very good at creating need. And so any attempt to educate the public is going to have to overcome the marketing acumen of a lot of very successful consumer product companies, and I wish you well.
Any other questions or comments, David?
I'm just wondering if there's anyone here who might have some thoughts about where this will take, where all these potential points of intervention may lead us with respect to the need for oversight and prudent regulation with respect to trials and testing and assessments. I know there are a number of people here that may have some thoughts about this.
Yeah, well, as I said, at least in terms of the fecal transplantation, which I think is a great test case and clearly a very promising medical intervention, their NIH and FDA are co-sponsoring a meeting to look at this. And, you know, we have seen probiotics, for example, that contain vancomycin resistance genes, other potential things that you might not want to get in your probiotic, particularly if you don't know whether it works for a given disease. So I think that this is one of these emerging areas where the trick is going to be to get the right balance between protecting patients but letting the field hopefully move forward. And, you know, clearly when you're talking, I think clearly understanding the underlying biology more, even in something like C. diff, in terms of, you know, as Josper was saying, what are the organisms that are responsible for the therapeutic effect, then you could have a defined product rather than feeding people stool, which would be, I think, a much better approach.
In the case report where they tried to make a mixture of 33 bacteria, they started with 62 bacteria, and then they selected only bacteria that were not antibiotic resistant, because I think that's indeed the major issue that you don't transmit pathogens, but if you are going to transmit antibiotic resistant bacteria, you can do harm as well. So I agree with that problem.
If I could just, and maybe it's a point of clarification, if some of the need is going to be for markers and endpoints that aren't numbers of specific named bacteria, but rather consortia with certain group properties, you know, net properties that can be satisfied by any of 13 kinds of mixtures, the names of which could be, you know, many multitudes more complex. That presents a different kind of sort of regulatory problem, because you're no longer saying we either did or did not have this organism, or did or did not see a fall in this particular, you know, defined, well understood endpoint, but rather, you know, used a quantity of property, and then saw an effect that is more functional rather than, specific species endpoint related.
But Jesse, reflecting on what you said a moment ago, not to prejudge anything that will happen at the May conference, but presumably, assuming that you can actually inventory the optimum package of organisms to be formulated into a pharmaceutically stable formulation, that's really the only way you could do a meaningful trial, presumably. The other issue, and I don't know if anyone has any perspective on this, what role do phages play in this relative to fecal transplants? And if they're excluded, is anything known about those transplants which have worked versus those that don't, as to whether there was actually a C. difficile phage population?
I mean, so, I think that the phage therapy, I see that coming back now as people, have run through all the antibiotics. And, but that's sort of at the beginning of the origin of this forum, and now I have to come back to those tools of, phage therapy and say, is that really what's controlling the bacterial community? I think, clearly it's the type of thing that, for Joost and I, we should be doing metagenomic sequencing. We should have the answers to questions like that. We shouldn't be just saying, we work 93% of the time. We should know what is the, full genomic complement that you're putting in. Because if that were true, if that's, if they're preventive bacteria or preventive phage, I mean, that's exactly the type of thing that we have tools for answering those, and that's exactly, we should be considering all of those issues when we're doing these kinds of cutting-edge therapies.
We don't know if there were non-pathogenic Clostridium strains that inhibited the growth of the pathogenic ones, but I think that's a strategy that is explored by others. Yes.
I just wanted to also say there's a simplification sometimes when we hear at least for me, when I hear, this sort of one-to-one relationship between the microbiome and a clinical effect. And, for example, why is it that a baby will not become infected, will not develop the inflammatory process, the inflammatory condition you see with Clostridium difficile? And I know what you're going to say, microbiome. It's not. You want to say it?
Well, we don't know that. It is still an interesting question, but there is a higher degree of carriage among very young children. That's recognized already for a long time, and some people think that maybe that's the moment that you can build up an adequate immune response that can protect you later in life if you are going to be an old cripple and attract Clostridium difficile after getting antibiotics.
Yeah, but yet there's another issue, and that is the mechanism by which Clostridium difficile toxin causes the inflammatory state, right? And this involves the activation of the nervous system. This involves substance P. I mean, there's a whole neurological cascade that is also part of that process. It's not a simple, trivial issue, and for reasons that I think it may be that babies don't have the receptor for Clostridium difficile, or some aspect of that cycle is not operative. And you would, with the discussions we're having here, leave the room believing that it's all microbiome. So I just want to say that the human body, physiology, is very complicated. And there's a simplification that happens, I guess, when a field is young, and, presence of this bacterium is causing that disease. But, the body is very complex, and you really have to put as much energy, granularity. You have to focus on the physiology, the nature of the disease process, how it unfolds. And to your point, it is a very complex, multidisciplinary effort. And I think if you stay myopic, you can run in the wrong direction. So it's only a statement that a condition like this, which is very complex, which can be presented in a relatively simple fashion to those who follow it and understand it, it is indeed complex with developmental, aspects and so on. And that's all. And you know what I'm saying. So that prevented him from comment.
Well, I think there's a really good historical perspective, because who would have been having this kind of conversation about the microbiome, 30 or 40 years ago, and here, but yet at that time they thought they were incredibly sophisticated, because we knew that there were B and T cells and we were going to solve all the world's problems. So I think, the microbiome is a perfect illustration that we are a living, changing ecosystem at all times, and that there's going to be a lot more to it than, but just like that led to treatments for disease, like rheumatoid arthritis, this could lead to treatments for disease. It doesn't mean we completely understand the disease, or that we won't get it wrong. Any other comments? And then we're going to, I guess David might want to close the meeting, or yeah.
I just wanted to bring up something that happened probably 10 to 15 years ago, where there was a probiotic that was being promoted for animal use, with the concept that they were going to seed the ileum and seed the colon to make sure that salmonellas and E. coli couldn't colonize in the animals. And one of these got approved at FDA, but one did not. And the reason being that they could not fully characterize all of the biota in the product. So again, it's the regulatory area may need to catch up a little bit to this process. You may need to bring them along. Thank you.
Is that a good thing or a bad thing?
I actually thought that you were going to mention it was a study that was actually in the New England Journal in the 1970s, the deliberate use of a non-pathogenic staph strain to replace Staph aureus in the nose. You remember this study? And kids got sick.
They died.
Yeah. Died of. People thought they knew what they were doing.
There was a nursery outbreak, and many children died.
Just to let you know how far we have to go, the current characterization of probiotics that I'm aware of is a 16S, and it's really just identifying that the supplement is what it's labeled. That's where we are.
Well, I don't have any great summarizing statements to make, other than it's of course, a pretty exciting time in this area of work, and it's made exciting by the fact that we have the means of discovering complexity that only offers untold possible explanations and mechanisms, but we don't know enough about any of it to realize that much of it may be wrong. So it's just that wonderful, sort of flush time in the early phase of a science before the reality sets in and we come back down to some reasonable level of balance. But nonetheless, I think there are a lot of wonderful questions that have been posed that beg, but also deserve specific kinds of studies that we can now do, and a lot of this can be addressed, as people have pointed out. So much to be done. Great models available, including people, lots of others, and I'm sure we will be getting together again in other settings to re-examine many of these questions, which have been addressed by many before us for many years previous. So thank you all again for a wonderful meeting, and look forward to seeing all of you again sometime soon.